today is going to be Joanna, once again, doing PCR and gel electrophoresis.
Yep. Uh-huh. All right. So what is PCR? I'm betting a lot of you guys have already done this. If you have, raise your hand if you've done PCR. Okay, you all have done it. Great. So polymerase chain reaction, lovely stuff. Do you know when it first happened or when it was discovered or created? Or by who? Anybody know either of those? A little bit of history for you. I was born, but I was still a baby. So let's just put that in context. So it was 1985, Dr. Cary Mullis. So anyway, he actually kind of, there's a really epic story that you can look up if you want, but he was riding his motorcycle out in the countryside, and it just came to him, the research that he was doing on something else would work for this. So it's one of those fun little stories. But anyway, so just a quick little refresher video.
Polymerase chain
just to
reaction,
remind you.
or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Okay, and this website actually has the video with Dr. Cary Mullis too that I was tempted to play for you guys, but it was really, he was a rambler, and I didn't want to feel like editing it. But if you're interested, this DNA learning group has it on there if you want to see what he looks like or hear him talk about his, you know, epiphany or whatever. It's not bad. It just was a little rambling. I didn't want to put it to you guys, but if you're interested at some point, it's not bad. And they have some other little activities and whatnot too. Okay, so just a reminder, refresher about DNA, because I'm going to be using some of these kind of terms, so you have to kind of remember what's actually about DNA. So we have these A, T, Gs, and Cs, and what are those called? Bases, yeah. Any other terms? Nucleotides. Mm-hmm. Yeah. So nucleotides is what I'm going to be calling them first, but bases is also correct. All right, so what about 3' 5' end? What does it mean to be at the 3' end? Do you guys know? Or versus the 5' end? Okay, generally, but what's it signify? What means, what does the 3' mean, like chemically? Yeah. Yeah, so that is important, the directionality of it as well. So this is just kind of a way that people kind of help you decide which direction you're going on things, and because there is a slight difference, and obviously it would be dramatically different if you read the other direction. And another thing noting is your A, T, Gs, and Cs, you can tell that they're different sizes. So Gs and Cs being a bit bigger, and this becomes a crucial point sometimes because, you know, it doesn't look much bigger, but, you know, your GC combo is bigger than your AT combo. And this becomes relatively important when you're talking on micro scales, which you will be when you're talking about PCR. All right, so what do you need for your PCR? So you need your ingredients, also known as your cocktail, and no, not that kind. Your equipment. To make it really basic, you actually do not need a thermocycler. Those make your life much, much happier. But I was talking to a older researcher colleague, and she remembers doing PCR by having several heating blocks and having to move it back and forth. So appreciate your thermocycler, that you do not have to move your tube back and forth. It was actually Linda that was telling me about that. And I was like, oh my God, I'm so glad we don't have to do that. So have appreciation for that. And you know, you need your supplies. All right, so just kind of your overview again. Need your sample, obviously. If you don't have that, there's really no point. But sometimes, you know, if you're doing this late at night, you may just forget or forget where you're putting if you're loading like a 96 well plate. So it's really a good idea to, like, you know, don't do this when your mind is feeling a little off. Just wait a little bit, you know, because there's that kind of trade-off between getting things done and getting things done right. So again, knowing yourself, if you're feeling a little tired, maybe it's not the time to set up a 96 well plate. Or if you do, idiot-proof yourself, put up a nice little map, like make a little chart for yourself and just check things off as you're going along. It's, you know, it never hurts to double, triple check yourself, especially if you're dealing with, you know, some very rare DNA that you have, or if, you know, you're using those really nice little PCR beads, which I love, but they're really expensive. So I don't think your PIs would appreciate it if you, you know, use one and you just forgot to add DNA, or you forgot to add, you know, your primers. That could be a problem. So you need all of these things in here. Depending on what you go with, sometimes you can get them all together or not. And you actually don't need tack. There are other options as well. So when it comes to polymerase, they've pretty much all been isolated from hot springs because it makes sense. You need something that can work at high temperatures. So tack is your classic one, which comes from Thermus aquaticus, which is a little bacteria there. But another one that is out on the market is PFU. So this is from Pyrococcus furiosus. So that's an archaea form. They're both from hot springs, and there's a slight difference in them. So if you choose to, you know, use tack, PFU will not necessarily give you the exact same thing. Just, you know, a heads up on that PFU definitely has its purposes. It tends to be a little more resilient than tack, but it doesn't always get the same results. So whatever you pick, make sure you keep that same one throughout whatever your study is. But I mean, tack is the one that's normally used, and there's so many different versions of it. Several different companies have made their own, you know, special tack or their special, you know, PFU, but those are what it came from originally. So these are just polymerases that have been isolated from these microorganisms that live in these really hot areas. So another really important thing when it comes to your PCR is your primer. So how many of you guys actually designed a primer? Did anyone? Ah, okay. Nice. So what kind of things do you have to take into account when you're designing a primer? So what'd you do? Did you remember? It was a long time ago. And it doesn't haunt you anymore? Okay. Reverse complement as far as? Mm-hmm. Right, right. So, I mean, you have to make sure that the code is the right. One thing that, you know, is an important thing that you can do as well is your melting temperature. So obviously it's really important to know, you know, what temperature that your primer is going to melt at. So usually, you know, and also the melting temperature of your DNA as well. So there's kind of a trade-off on these. So usually, you know, it's like, whew, trying to look at my notes and it just scrolled away on me. Again, this is where your GC content comes into play. So generally speaking, you know, you have your, if you go back to our lovely thing, so you've got your denaturing at 95. So 95 is usually a good, you know, one to get your DNA to separate. But if you have a really high GC content, it might be higher, or you might want to consider going higher because it might be harder to melt it. But generally, it's right, but I mean, a lot of this is how much do you know about your target, because obviously you need to know the sequence of that, depending on what you're going for. And so if you know it has a lot of GCs, you might have to go a little warmer. Or you might, if some things might be more sticky. There's a lot of different things on it. Or hold it at longer is another option. So we'll kind of get into those in a little bit, but there's different ways to play with PCR. I mean, and you guys are probably lucky that somebody just gave you a protocol and you just said, oh, lovely, I don't have to think about it. But it's sometimes nice to actually know what's really going on and how you could tweak it to maybe make it better. So again, melting, and then your annealing temperature too is also important. So usually primers, their melting range is normally from like 52 to 58 C. But you usually want to be around 3 to 5 C below your melting temperature to get things to operate correctly. But when you're calculating it, so your temperature melting is 4 times your GC plus 2 times your AT is kind of a way to estimate it. So again, your GCs are weighted heavier because they're harder to break apart. So obviously the length matters. A longer primer is more specificity, but also you risk your primer shearing and falling apart too. So there's kind of a lot of trade-offs with this. And your amplicon itself, what your target is going for, the length of that too. Because if it's too far between the primers, you risk it, not being able to grab it very well. Or, if it's really close, there might be some mismatching. And sometimes you actually do that on purpose as well. So another thing is you want to avoid these runs because, if you end up with having, too many of the same in a row, you end up like sliding your sequence often because it'll bind to part of them but not the other, and it kind of will shift and slide. So if you have like something like this within the sequence you're going for, maybe you should slide your primer to one side or the other side of this region because you risk having these kind of sliding matches if you have these runs. So, for this example right here, your base G has a value of 5 and 4. So you can see that 5 run and then that 4 run. So you really want to avoid anything above a 4. Like 4 is kind of okay, but if you go with a 5 run, it's very, very risky. So when you're looking for that ideal spot to plunk your primer, try to avoid those areas. And also you want to avoid repeats, the same reason, that same, that AT, you're risking having, again, that sliding mismatch. Okay, so sometimes you want to allow a mismatch. So there are things that you can call degenerate positions. So for example, you can create an artificial nucleotide called an M, which will accept either an A or C, will act as an A or C, and a K, which will act as a G or a T, and so forth. So you can create these, artificial nucleotides that will accept one or the other, or 3 different options, or be completely degenerate and bind with anything. So this can be helpful if you don't know exactly what your target is, or that within the group that you're going after, it may have some differences and you're okay with that. Maybe you want to get all that's of this type, and so you're going to allow that little mismatch within the primer because maybe you're looking for a group of related but not identical, and they sometimes have a little difference, and you're going to allow for that little mutation or whatnot. So you can create these artificial nucleotides that will accept a slight variation or accept anything. So these are obviously ones that do not exist in nature, but they're ones that we can make in the lab. So you can order a primer that has these degenerate nucleotides, which can be kind of fun to play with, but also can drive you insane. So there's a fine line here between, being, it's kind of a thing that's really great to do all these little tweaks, but also you could spend ages and ages just, substituting a different nucleotide or, creating a higher melting temperature by adding more of something or less. And yeah, so there's a lot of ways you can go with this. It's basically the end story, and people have. So when it comes to your mixology, you can make your own. So often it depends what your lab you're in, how they like to do things. Some of them like to control everything. So that means you add your own nucleotides, and you can actually buy separate A, T, Gs, and Cs as well. If you want to bump your ratio, you want more A's and T's in there, you want more A's, you want more T's, you can do that. Buffers normally is kind of a tris salt buffer, but you can add to that as well. There's different additives. And of course, the polymerase, you've got, your two options, two major classes of options for polymerase. Or you can buy your nice premixed. So again, not those ones, but those ones. So like I was talking about the nice little beads. The beads are really awesome because within it, it already has your polymerase, your buffer, your nucleotides. All you have to do is add your primer, your DNA, and some water to get it up to concentration. So generally speaking, you have to add water anyways. Water is often the first thing to get contaminated to. So, often when you buy your really ultra pure, super awesomely clean water, you aliquot it out into little tubes and freeze them, or everybody gets their own tube so that if something goes amiss, you can't blame it on someone else. Usually a good thing to do. Often with PCR compounds, people like to have their own anyway, just because, Joe Schmo in the lab might not be as clean as you are, and, darn it all, if he uses your stuff, you can't trust any of the PCRs after that. So it's nice to have your own. But the good thing about these, little tubes is that it comes with this way, but the bad thing is it's harder to tweak all those little differential things if you want to get into that, which you definitely can. So then when it comes to these additives I was talking about, the most common one is magnesium chloride. So usually this can affect the specificity and the efficiency of the reaction. So higher concentrations of magnesium chloride, so it'll have lower fidelity, so it's more apt to accept, which can be good for you if you want to allow that wiggle room, but also can be bad because you could end up with a whole lot of unintended bindings. So it's trade-offs of how much do you know about your target and what are you willing to accept. So normally like a buffer I think comes at like 1.5 millimolar concentration, but often PCRs can be found from 3 to 5 millimolar depending what you want to do. So another common one is DMSO, so dimethyl sulfoxide. So this is particularly useful if you're dealing with a GC-rich template. So it can reduce your Taq polymerase action, so it kind of allows you to get in there and get to those before they kind of suction back to each other, which sometimes happens, or they didn't break apart or whatever. So sometimes it can be helpful to add that DMSO. Another common one is betaine, which yeah, I know. I had to look up on how to pronounce that because I've never used it, but a lot of people do. And it's just kind of one of those general, hey, we added and things work better. The exact mechanism isn't exactly known, but it's one of those things that people like to add and, hey, it works, so okay. And kind of the same thing with these non-ionic detergents. So a lot of those are classically sold and created by different companies. They will say, we have this, PCR clean additive you can add and it'll make your PCR better. So it's usually just some sort of detergent. So, the thought is that it kind of suppresses the formation of these secondary structures. So basically your DNA kind of loops up and binds to itself. And so if you have like maybe a large target zone, it might be a good idea to add this because, your thing's floating around and it may try to bind with itself instead of being a nice, beautiful template. So there might be a need for using these. There's also TMAC, trimethyl ammonium chloride, which is another fun one. So this is supposed to help eliminate non-specific priming, and it's also been used to help reduce the DNA-RNA mismatch if you're doing a reverse transcriptase, so if you're going from RNA to DNA. It's supposed to help with that. And my personal favorite, BSA. For some reason, this was the one that really helped me. So it's bovine serum albumin, best if you don't think about it too much. But for some reason, I found that this really helped my PCRs when I was dealing with really dirty samples, and I mean that literally. The sample was lightly brown colored, had like a lot of tannins in it. It was like water from the ground, so hyporheic water from the river beneath the river. But yeah, so I found BSA worked really well, but if I think about it too much, it creeps me out because you can guess from the name where they get it from. So it's from, pregnant cows. They get it right from the, fluid there. So bovine serum albumin. but it works. Sorry, which what? Yeah. But for some reason, it works miracles when you're dealing with, like, inhibitors. And yeah, don't know why. And it also works really well if you have like ancient DNA. So if you're trying to PCR some, long dead organism, for some reason it seems to help stabilize it too. I don't know, maybe it's like, from the womb, there's all sorts of magical chemicals that help. And again, it's like a mixed thing. It's not a pure, and like people aren't exactly sure why it helps all the time, but it really does. Weird, but true. Okay, so also you have your other options. So with the thermocycler, you can decide, how many cycles do you want to run. So it depends, how many copies you want to make, because, more rounds, more copies, but also more chances of error, depending on all those little factors that you did when you designed your PCR. And again, you can change those temperatures, within a range. Obviously, if you go way beyond or way below, you're not going to get the action you want, whether it's, breaking the DNA apart or activating your polymerase or deactivating your polymerase. So, there is a range of acceptable with, any enzyme or any physical reaction. So sometimes you can tweak those little temperatures just a little bit. And of course, having your positive and negative controls. Has anybody forgotten to include those on a PCR before? Because I have. And it really sucks that you, did your nice PCR and like they're like, hey, look, it amplified. And your PI says, well, what are you comparing it to? Oops. So make sure that you have a positive, that's something that should amplify based on your primers and your PCR conditions. And you should have a negative too, which includes everything but a template DNA. So make sure you have those. And if you are tweaking with conditions, then it might be nice to have a control for each one of those if you want to try to optimize your PCR. So maybe one that, has a higher magnesium chloride concentration, one that has lower, or one that has BSA, one that doesn't. So all those different, cocktail recipes, you can make a different control for each one of those as well, if you want to go down that rabbit hole. So again, duplication, replication, always a good idea when you're doing research. So, if you're running some sort of rare sample, then maybe you won't do a whole lot of replication, but sometimes it's a good idea, especially if you're doing, low cycles and you want to make sure that it's, working, but you don't want to, extend your cycles. You want to still have like, err on the side of the pure, but less copies. Maybe, you want to do it more than once, that same sample, just to say that, it's definitely valid, that it's definitely going to create enough that I can take it to whatever downstream process I have in mind. If you're taking it to sequencing, you obviously need a certain concentration of DNA before, the sequencer is going to be working efficiently. I mean, you can run it with lower, but it depends what sequencing technique you're using, what concentration they suggest. And again, there's a range, but, if you have more, that's pretty much better when it comes to sequencing. So if you have a really low concentration, you might be kind of rolling the dice whether it's going to successfully, amplify going into the sequencer. And meticulous on the front end. So before you set up your PCR, especially if you're doing microbial PCR, you want to be very, very tidy with it. So a lot of times labs will have sections of their lab that are pre-PCR and post-PCR. Or they'll have, dedicated pipettes that are, for prepping PCR. Or again, if you have a lab that has the funds, you can have your own set and at least you know what's been happening with those. And, using definitely clean tips. Don't use a partially open box unless you're the one that opened it and you know what happened to it, or you have that dedicated tip box in the hood that is only for PCR and you trust your other lab mates. No judgment there, but, be honest, because you don't want to set up your PCR and find out that it didn't work, which happens, but try to err on the side of awesomeness. And, obviously surfaces too need to be cleaned up. So usually, maybe some bleach or some ethanol. So 10% bleach, 70% ethanol, do both. Why not? It won't hurt. UV is also another one. So if you have, in the hood, you've got a laminar flow hood, not a regular flow hood, because you don't want the outside air going out and over you. So you need a laminar flow hood, or don't use a flow hood. You can do it next to a flame with an updraft. That'll help keep it relatively clean too. Laminar flow hood, obviously the gold standard, but don't do it in a regular chemistry hood, because those are meant to protect you but not your sample. So they blow in air from the outside and just shoot it up. Yes, we've had issues with contamination from those before. So again, you know, the air would drop things down into your sample too. So be meticulous when you're setting these up, because DNA is everywhere, and things that will, you know, get rid of your DNA are also everywhere. So, you know, try to be really clean about it. Another common thing that happens is ice. So this is probably more if you're making your own, because if you're making your own cocktail, you've got all these different things, especially the tack is very sensitive to degradation. So you're, and same with the PFU. So you want to keep those really, really cold until right before you need them. Some people never take them out of their freezer except for just to, you know, squirt it in the very last thing into the cocktail. But usually then they keep their cocktail on ice too, and if the tube, you know, kind of tips a little bit and that ice gets in there, that's a big problem. And I know that that has happened before, which is why some people use freezer blocks too. But, so those are sources of frustration often. All right, so any questions about plain vanilla PCR? All right, so we're going to get a little fancy now. So one common one is called multiplex PCR. So this is basically, you know, you have a unique primer set for multiple different targets, and you can, you know, throw it all in together, making sure, of course, that these are uniquely different. Or if you're using it with qPCR, which we'll talk about later, you can put different fluorophores on these sometimes, depending on the capability of the equipment you're working with. So you can have a different fluorescence attached to these. So basically, yeah, I mean, I wouldn't do more than 4 generally in one, but people do sometimes. But usually 3 to 4 is probably as much as you want to multiplex, just because it's really hard to have unique primers depending what you're going for, again, that aren't going to overlap, or there won't be any mispriming, or, you know, say that, you know, primer A could bind to primer D or something like that. You end up with primer dimers, which is really frustrating as well. So got to be careful what you choose, but it can be really useful for certain things. So I know it's used a lot of time with, you know, SNPs. People familiar with SNPs? Is anyone not and wonder what I'm talking about? Okay, cool. So another fancy one is nested PCR. So this is usually 2 different sets of primers used in 2 successive reactions. So in your first PCR, you know, one set of primers is used to generate a DNA product with may or may not contain certain parts from your targeted areas, and then you do another PCR that kind of gets at the other part of it. So you end up with, you know, this unwanted product happening, but eventually you kind of get down to your targeted sequence. So you could do this if you have, again, like maybe some kind of sloppy areas in your target that you want to avoid, or you want to maybe narrow out certain parts. So you'll do your first primer to pull out a big chunk that you're interested in, and then you're going to go in again and get more specific. So there's different reasons why you might do a nested, but there's a lot of people that do. And then, of course, there's reverse transcription. So again, polymerase won't work on RNA. So what you do is, you know, when you're making your cocktail, you also add your reverse transcriptase. And so that's going to create cDNA. So, you know, cDNA is kind of like if RNA could be DNA. So it's basically double-stranded RNA in a way. That makes sense. Anybody made cDNA before? So much fun, guys. All right. Now, here's an area that's often confusing. So I usually consider reverse transcriptase RT-PCR, but there's also real-time quantitative PCR, which I call qPCR, but sometimes people still call it RT-PCR, or RT-PCR, which will totally mess with your mind. So just be aware if you read something that says RT-PCR, they might mean real-time, i.e. qPCR, or they might mean reverse transcriptase. So just keep that in mind, that there's different naming conventions with that, and it can be a little confusing if somebody wasn't really clear up front on that, which is why I'm not going to call this RT, but some people do. I'm going to call it qPCR. So basically it's PCR with fluorescence. So there's 2 major types. There's your non-specific that just binds to any DNA. And then you have your sequence-specific DNA probes that are labeled with a certain reporter dye, which is only detected when a probe connects with the target and the suppressor goes away and then it shines. So that's kind of a little different way of doing it. So there's two major types here, essentially. So sometimes, you know, it's good to run both of them. Sometimes people only like the specific one, but of course that's going to be more expensive because you have to make special fancy primers that are specific with this. I mean, you still have to as well with the non-specific, but it's a lot easier because you just have that lovely little cyber green dye in there. So these kind of qPCR in a way can eliminate the need for gel electrophoresis because you're getting an idea, because every time a copy is made, it flashes essentially, and your fancy qPCR thermocycler records that. And so you kind of get an idea as well. So you sometimes, I mean, generally when you're doing qPCR, there's no need for taking it to the next level and going to gel electrophoresis. However, you know, qPCR products, depending on what's going on there, it might be really difficult to take it to a downstream option. So you wouldn't necessarily be able to take these and then send it off to a sequencer. You'd have to go back to your original DNA. So there's, you know, pros and cons to it that, you know, qPCR gives you this really great data, but it's also sometimes confusing to read it because you have to do things with melting curves and, you know, a lot of quantifications. Has anybody here done qPCR? Is it, how did it go for you?
It didn't go well.
Even with Ian there?
It wasn't with Ian. It was when I was in Rawhide and I was with you for
Okay.
and he scrapped it. Yeah, it did not go well.
Yeah, qPCR can be really great when it works, but it can be really difficult when it doesn't. So in a perfect world, you know, people would pretty much only be doing qPCR. But because it is frustrating with all these little, making sure that your primer is very perfect because it has a low tolerance if it's not. Whereas, you know, if you were with your regular PCR and it wasn't priming perfectly, you know, you'd still be able to see something. But because you get your final product, you don't get, you know, these flashes each time a cycle goes. So, yeah. I have dabbled with qPCR and I've been pretty frustrated by it in general. So I know it's a wonderful tool, but it has a lot of little caveats to be able to actually get it working well and troubleshoot it and trade-offs again. So if you didn't do qPCR, you're probably going to move on to gel electrophoresis. So all of you guys said you've done PCR, so I'm assuming you all have run gels? Yep, okay. So I'm just going to do a quick little overview that, of course, you know, you've got your lovely gel, you know, you load it up, all about electricity and staining. All right, so let me go back to that one. So what side do you load your sample on? The positive or the negative side? Huh? Yeah, so it's the negative side, right. And why is that? Perfect, yes. Which, you know, I've before, you know, connected the leads on the wrong way, and all of a sudden, this is why you add dye to your sample as well, because you see the dye going back and you're like, oh my gosh, quick, switch the leads. And it's all good. But be aware of those kind of things, because it happens. Especially if you're working, you know, late at night or you're tired, so be aware. It happens and it's okay. And then, of course, you know, staining here. So kind of your overview again. So we kind of covered most of this. So longer fragments are going to go, what, faster or slower? Slower, yeah. So they usually get caught up at the top. So that's why running just whole DNA is not really useful generally, but sometimes you do it when you're teaching somebody just how to load a gel, because basically it gets all clumped up at the top, and, you know, unless you're running it for a very, very, very long time, it's not going to move very far. And so again, you know, it's important that you have something to compare it to. So you have your ladder or standard. And there's various different ones that people use, but again, it's just making sure that you know the sizes on it so that you can use it as a ruler essentially to measure over. So did you guys actually, when you did your gels, did you measure over from your ladder, or did you know what you were getting and you just said, hey, I got a band, it's got to be it? Okay. Did everyone measure? Mhm. In a perfect world. But I know sometimes that doesn't happen. When I was first taught, I did not get that information till later. And so, the person who was teaching me didn't give me the full information. So it was like, we knew what we were looking for. If you got a band, it's it. If you didn't get a band, it's not it. But it is really important to know what size you're looking for, and that's the strength of having the ladder too. The people I was working for originally, they just, didn't give me that, and they said, well, this is the primers we're using, this is the size it's going to get, so if that's what you got, then you got your product. But that's not really a good way to go about it necessarily, because there could be several things that happen to be that size. But so that's why I use the ladder to measure over, to kind of get your lovely idea there. So again, options, options, options when you're making your gel. So when you guys made your gel, what did you use as your buffer? TAE? Anyone use anything else? Okay, so you could also use TBE. So the difference with using a TBE is it tends to be better at preserving things, which can be really great if you're running a long gel, but it can be a bad thing if you have any downstream processes. Like if you wanted to cut out a band, you would not use TBE because it binds things up so nicely that you would not be able to use that for cloning or sequencing or anything else. It's just nice and clean, which is great, but not so great if you want to do anything else with it. And it's also a little more expensive. So usually people use TAE, but you can also use TBE. It does have a really good strength if you're running a long gel. So if you have really big fragments and you want them to be really nice and clean, so you have low voltage in a long run, you end up with something a lot nicer from that. Because your base goal is to get a good picture. So gel concentration is another really important one. So out of curiosity, does anyone remember what concentration of gel they made? 1%? Anybody else? 1%? Okay. I mean, usually that's the standard. It's easy to calculate. Yeah, I mean, there's a lot of variability here. And so if you use different concentrations, you get different pictures. So these were all run at the same voltage, but these were cut out and added together. But you can tell that, on this more concentrated, you end up with this kind of, smearing, that things move slower. But if you use a lower percentage, it's easier for things to move through it, which makes a lot of sense. If you make it thicker, it's harder to move through. But it also makes the gel more stable. So usually, if you do a more concentrated gel, you usually have to do either lower voltage or longer run. Because those are the factors that you can kind of control with that. There's also, of course, the dyes and the stains. So the dyes that you actually load it with. So usually the normal ones that people use are xylene cyanol and bromphenol blue. It's important to note the general size of these as well, because they can kind of get in your way. If your product happens to be the same size as your dye, that can be a bit of a pain to visualize it because you end up with that nice little dye band on there, and it's really hard to see your product. So being aware of those kind of things and choosing an appropriate dye can be important. And of course, your ladders and standards. And again, current and run time. So I mean, there's various different voltages, and it depends also how big your gel is. Because of course, if you got a bigger gel, it's going to take it longer to run than it would if you were running on a smaller. And higher voltage, usually it makes things run faster, but you also can get a smeary picture. So it's usually better to run a little slower because you get a better picture in the end. So, run a little lower voltage and it's going to be a little longer. And again, if you use the TBE, you can let it run into your gel and then you can fridge it overnight and put it back on to run. So that's another benefit of the TBE, is if, you started it, you ran it, and you looked at it and you're like, I think I'd like it to separate out a little more. It's completely stable to sit in the fridge and wait for you tomorrow. Whereas TAE probably will be, but it might leach out a little bit. So again, your trade-offs there. What was I going to say? Oh yeah. Some other little random note that I was going to say, but I just completely forgot about it. Oh yeah, dyes and stains. Okay. So the other thing with stains is your post-run stain. So how many people used ethidium bromide? Okay. So ethidium bromide, very efficient. You're a nice, historical one, but also a known carcinogen. So a lot of people are moving away from using ethidium bromide and using a lot of these more expensive but safer alternatives. So there's like Gel Star, there's Cyber Gold, I think. There's a couple of different companies that make them. And the benefit of some of those, and you can also do this with ethidium bromide too, is actually put it in the gel itself. So when you've melted your gel and it's, at the there's no floating particles anymore, you can add your dye to it at that point and gently swirl your dye into the gel itself. I would not recommend doing that with ethidium bromide because then you've contaminated more things with ethidium bromide, but people do it. But a lot of times if you're using one of those alternatives, it's a great idea to put it in your gel itself as well so you don't have to deal with the staining step as much. I mean, you probably still will, but you've kind of got yourself a leg up if you put it into the gel itself. But again, don't do that while it's still hot because then your dye is going to break apart and that's unfortunate. So you want to do the kind of baby bottle test, with your gel to make sure it's not super hot anymore. So if you're not familiar with baby bottle test, just put it up against your wrist, see if it feels hot to you because that's a nice sensitive area of yourself so you can see if it's good to go. And of course, your normal post-staining setup, generally, in a little tub and, who knows how long that gel mix was made, so you can always respike it with dye if you don't know or if you don't have a little sign up saying when it was last spiked with dye. So things to be aware of as well. All right. So your overview. All right. So thanks for listening to me. And now you're going to get a lovely little scenario. Two extras in here. So if you got one of the really easy ones, you might have to get another one. But while you're thinking about that for my own entertainment, the PCR song.
There was a time when the amplified DNA
You had to grow tons and tons of tiny cells.
Oh, and along came a guy named Dr. Carey Mullis.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primer. Nucleotides and polymerases too.
denaturing and healing and extending. Well, it's amazing what heating and cooling and heating will do.
To all the